Welcome everybody to our webinar of today. As you may know, we have our bottom bar where we can have our Q&A. You can do all the questions you want. You just need to push the button down there where it says Q&A and you can type your question and we will answer it at the end of the webinar. We have also one option, which is translation. So if you go under and you can find like a circle down there where it says interpretation, you can have the Spanish version of the webinar. We will also be recording the webinar so you can repeat it later, and we will send out the PDF version of it so you can have it on your library, in your incubation library. Feel free to ask any questions. So today we have our manager of consultants, Dr. Keith Bramwell. Welcome.
Good to be here on this side of the screen.
Yeah, it's weird, you know, like having you're always on this side, so it's kind of funny to have this different one.
Yeah. So we ready to go then?
Yep.
Okay.
Sure.
Let me share my screen and see if I can do this correctly. All right. Perfect. Got those kinks worked out. All right. Thank you much. It's good to be here. Like I said, it's different being on this side of the screen, I say. Carolina, not whether she mentioned our normal translator is not with us currently, had some emergency come up, so Mauricio will be doing his best as far as the translation if you go into the interpretation box. So anyway, with that, I will go ahead and get started. So I'm going to talk about some hatch residue analysis, and I've talked on this a number of different times. Actually did a webinar on it about 3 years ago, I believe, last I looked, and so it's been a little while. This is going to be similar to that, a few little changes here and there. Try not to overstep on some other speakers we have, but we'll go through this and talk about it. And a lot of it is I'm really big on, whether we're talking about incubation or farm or whatever, is letting your chicks talk to you, letting your birds talk to you, letting your results talk to you. But part of the results are the chicks themselves. So letting them talk to you by their growth, development, and their status in our cycles. So when we look at incubation, there's a lot of different management guides. Okay. A lot of different management guides, but one thing we always have to remember is that these are guides. Whether it's when some of our primary breeders that we deal with or ourselves, these are guides, and we need to utilize these as such. Incubation is a science, so we know exactly what needs to be done for the most part, but is also an art. That's where we have to adjust. A hatchery in Egypt and a hatchery in Nova Scotia, Canada, they have such different external environments that we do have to start making some tweaks and changes based upon our results. And the results that we're going to be looking at today from our hatch residue and our chicks, and there's a lot of other things. I'm focusing mainly on the chicks and the development, and will give us an opportunity to make some decisions to get into that art of incubation side. So real quickly, the basics of incubation. And I'm not going to spend a lot of time on this. We should all know this forward and backwards, I would think. But as a reminder of it, the basics of incubation, there's 4 things. Temperature. It controls embryo growth rate, faster or slower. So if we have a higher temperature, we will typically have faster growth rate. If we have a lower temperature, we'll typically have slower growth rate. That could be temperature within the entire cabinet, or that could be in microenvironments within the cabinet. If our ventilation's off, we may have some warmer spots and cooler spots, and then we get slower and faster growth of our embryos. And so we just need to remember that, particularly at the end when I start talking about some hatch, analyzing hatch and the status of our hatch. We need to kind of go back and say, okay, so what's going on here? What could be causative factor of this? Does that mean that we can just keep cranking the temperature higher and higher and higher and get the chicks out earlier and earlier, or turkeys or whatever they are? No. And without going into a lot of the embryology, they just need that time to develop properly. Much like a breeder hen, we can't just put, when we can physically, we can push them with growth rate and get them in egg production much sooner than we normally do commercially, but it's not very effective. The birds, we don't have the performance production out of them that we should, because we didn't allow them to grow mature like they should. So temperature, humidity controls moisture loss. And that is important. I'm not going to talk about that, moisture loss and humidity in the machines, but that's one of the other basics of incubation. Ventilation, gas exchange, temperature, moisture exchange are affected by this, and the uniformity of both. And then turning for embryo development, and also to it changes the environment within the cabinet a little bit when we do turn those eggs, and it redirects our airflow throughout our cabinet. So anyway, basics of incubation. Today, most of our successful hatcheries have really realized that, the world's changing, and I'm never going to say the genetics are worse, but they're more fine-tuned. And we know in management, I spent a number of years in academia at University of Georgia and University of Arkansas, studying a lot of breeders and hatchery stuff, and we know that it is more difficult to raise some of our replacement breeders now than it used to be. It doesn't mean they're worse. It just means we have to evaluate what's going on. And I think the same goes along with incubation, is we may be able to cut some corners previously, and we really can't do that anymore. And I don't think it's going to get easier as time goes on. So we have to pay attention to the details to be able to get the most out of our hatcheries. Looking at the guidelines, looking at recommendations, and I say this as the director of consulting, we send a lot of our consultants out to a lot of your hatcheries, and if you do have any needs, just let me know. But we sent to a lot of your hatcheries, and we give some recommendations, and these recommendations come from years and years of experience. We're talking with somebody that's maybe been in 100 different hatcheries in the last few years. They've seen a lot of things, and so paying attention to the recommendations and the guidelines set for, but then also be willing to make changes if we need to. One of the things that we need to, I always say with hatcheries, is to get good, successful hatch, we have to have a good breeder program. This shouldn't have to be said. You cannot hatch an infertile egg ever. Unfortunately, we have to say that, because we get customers that come and say, hey, we're just not getting a hatch. And so, well, you're also down 2 or 3% in fertility. Well, that's your hatch. We can't hatch eggs that are not fertile. Also, when we have reduced fertility, primarily due to infrequent mating, we get, of course, reduced hatchability. We get lower percentage of hatcher fertile, meaning we get a lot more early deads and some other late deads as well, because we have an embryo that's developing, but it's not as viable. And this goes way back in some literature, back in even the 40s and 50s, where they called it old stale sperm was the term. That if hens are not mated frequently enough, and they're having to utilize the sperm that they're storing in their bodies too often, then they can get fertility, but they get less viable embryos and chicks. Chick quality also stands a reason. If we have some embryo mortality issues due to fertility drops, those chicks that hatch were also stressed. For one reason or another, they're the same situations applied to them as far as fertility and the spermatozoa used. So we get reduced chick quality, we get poor broiler performance. And I did a lot of research on this when I was in academia and looking at that, and it held true every time we looked at it. Bottom line, you can have a great breeder program and a bad hatchery, but you cannot have a bad breeder program and a great hatchery. So that kind of just gets back to the starting of everything. We've got to have fertility. We've got to have fertile eggs. That is the fuel that goes in our machines. We've got Jamesway, Chickmaster, incubators, both very good, very, very good quality incubator. A lot of research time and energy in the development of these machines and the equipment and how they work and how they operate. But we've got to have good fuel, which means good eggs, good quality eggs, not just fertility, but good egg pads. So we got to have a good program and have everybody work together. Looking at fertility itself, the fertilization process, and I've talked about this many times, but I will touch on it a little bit again. That's a whole process. So once the hen is inseminated, either artificial or natural insemination, That sperm then is stored in the hen, and then it's moved to the site of fertilization. It transports up the oviduct, and then recognizes binding sites and undergoes an acrosome reaction, and then we get actual fertilization. I'll touch on a few of those things a little bit. Where does the fertilization occur? In the upper portions of the oviduct, near the infundibulum. So essentially, a hen walks into the nest. When she sits in her nest and lays an egg, the next ovulation will occur 20 minutes later. Very close to that 20 minutes. I've done a lot of work with this, but 20 minutes later, she will lay the egg that will be laid the following day, usually. So when that ovum is released, it is captured by the infundibulum, and it starts its path down the oviduct. The fertilization itself will occur at that germinal disc area. The little white, this is an infertile one, but that little germinal disc area, that dot that we see in there, that's where fertilization and embryo growth and development will occur, in that spot on that egg. During that fertilization process, what's occurring here? We've got, the fertilization occurs within 5 minutes. The ova is then present in the infundibulum, maybe 15 minutes, but within a few minutes, we're getting albumin already wrapped around that yolk, so fertilization process stopped. Once we get any albumin on there, there's an inhibitor in that albumin that inhibits that acrosome reaction and the sperm process of fertilization. So then we move on to the magnum, where the first layers of albumin are added. They're about 3 hours, passes down that oviduct to the isthmus, and the inner and outer shell membranes, and albumin are added, plumping, what's called, to fill up, to get water to that albumin, that dense albumin that's there. And that occurs about an hour and a half. It's in that portion, and then it goes to the shell gland, and it will be in the shell gland, roughly about 20 hours, getting the remnants of that shell formed. These little areas right here that you see, these little bumps, are the sperm storage tubules. I'll talk about that in just a little bit. So that's where the sperm is stored within the hen long term, yet fertilization has to occur up here. So we have to have a transportation path or area for that sperm to move. So then once they get to the uterovaginal junction, the vagina, they're there just a few minutes until the egg is laid. During that shell formation, it takes about 24 to 26 hours. The body temperature of the hen is 104, 105.5, or 40 to 41 Celsius. And of course, that temperature is warm enough that we get embryo development. So when that hen lays the egg, we may have anywhere from 40 to 60,000 cells in a freshly laid egg. And that's before we even start the incubation. So it kind of helps us remember, I'm not going to talk about egg storage here, but this egg we get is already fertilized, and the embryos already started growing at the time that it's laid. So, it's not like a fertile egg that we're going to then put in a machine and start embryo development. It's fertilized and the embryos already started. So then we kind of hold and suspend them at that point to try and get all of them in a similar fashion. So when we look at an infertile and fertile egg unincubated, we see the infertile egg is more of a close compact dot, and then the fertile egg is more of a donut shaped, and we'll talk a little bit about that. This will be covered a little bit more in our next webinar, February 14th, Valentine's Day. Dr. Michelle Beal from Select Genetics is a veterinarian with Select Genetics, very, very good, very experienced, a lot of good experience. She will be presenting a webinar mainly from the turkey perspective, but looking at fertile and infertile and candling to assess reproduction characteristics and some other things in there that you don't want to steal her thunder, but she will talk a little bit more about this then. But we see in that unincubated egg, we do see that white dot, that germinal disc, and we see it start to grow if that egg's been fertilized. Talked about the sperm storage glands. So again, in that oviduct, these little crypts or areas in that uterovaginal junction look like this, or this is an actual electron microscope graph of that. The blue in here is are the sperm cells that are lined up in there. And then so if the hen is mated artificially or naturally, and those sperm storage are full, they will continue to empty out to keep that hen fertile, so she doesn't have to get mated every single time she lays an egg. If we go several weeks without mating, they'll start pulling more and more sperm that's been in there longer because it's last in, first out. So this sperm at the bottom here may not ever be used if there's a lot of mating activity going on. If not, then we start having to use this. It's old, and that's where before it basically dies, that's where we get some mortality problems. So that fertilization process, the sperm attaches to a paravitiline layer, releases enzymes, digests a whole, and gains access to the site of fertilization. In the avian system, we can have multiple sperm enter, but still only one fertilizes, different from the mammalian system. If any of you have gone through that, studied that, which I did a lot of both, it is very different. We can have multiple sperm there. And going into this, there's a test that I developed a number of years ago where we can look at the sperm penetration, the actual occurrence of the sperm penetrating. This would be, and this can be done in a hatchery, it's pretty simple with a laid egg. All these dots then will represent where a sperm cell is bound and acrosome reacted and passed through to the germinal disc to fertilize. So this is what we like to see, a lot of holes here. Again, only one shares the genetic material, but it indicates there's a good mating activity. So we get some like this, we get some here where we only have a couple of holes there. That does indicate something. It's a whole nother discussion I can have with people, but we have a method that we can actually measure, other than just is it fertile or not. We can measure mating activity and the presence of sperm in the hens, and they're available for fertilization. And developed in a lab, but it's been a little bit simplified since then. So again, looking at our infertile and fertile eggs and unincubated eggs. So the blastodisc, if it's an infertile egg, once it's been fertilized, it's a blastoderm, meaning, okay, now we're going to start growing an embryo here. After 12 hours of incubation, roughly 12 hours of incubation, we can see that small donut we saw here starting to grow bigger, and 24 hours it's bigger yet. And if we were to continue to let this grow, you can kind of see the pattern where that is going to grow. That area here will completely envelop the yolk. It'll come all the way around, and that yolk will end up being inside the chick when it hatches, the remnants of it. So this is what we're looking for, is organized growth. Now, sometimes if you're doing embryo breakouts and you see some and it's just a mess, I'm always looking at this, is there an area of organized growth around that germinal disc? Because that's a sure sign that we had fertility and a start of an embryo. And it may not even get nearly to this point, but if we see that pattern, whether it be smaller like this or more developed like this, I've honestly seen in other countries where they don't have access to on-farm egg cooling in incredibly hot parts of the world. Even their transportation trucks are not well. I've seen eggs getting into the hatchery that are unincubated that almost look like this. So they have had a significant amount of embryo development growing. A couple of problems with that is, one, it creates an ununiformity. So if we've got some eggs that essentially have had maybe 18 hours of some development occurring before the incubators and some without, we're guaranteed right there we're going to have some ununiformity in the hatch. It's one of the reasons why we want to store eggs. One, to keep the integrity of the egg yolk, albumin, all that in place so we can grow a good chick, but also we're trying to maintain that uniformity. Looking at the relationship between fertility and embryonic mortality. So this graph here is some work we did a number of years ago. We inseminated hens with 200 million sperm. It's estimated that in a natural mating, between 100 and 200 million sperm are deposited by the male, depending on his mating activity. End of a long day, it's much lower, but 100 million is what's considered where we want to be. And so in this study, we inseminated hens. We followed them out after a single insemination. So we can see fertility here after 8 days after a single insemination. We see fertility start to drop. And we will see this, if they're only inseminated once a week. But after that, we see a very rapid drop in fertility on day 10, 12, 14 after a single insemination. But what we also see here is this is the percentage of fertile eggs that died early embryo mortality. So really 12 days out, we've got almost 10% of those fertile eggs are not going to make it. They're going to die in the first 2 to 3 days of development, because they've had to utilize sperm that's a little bit old and what we call that stale sperm. So they have the ability to fertilize an egg, but not necessarily produce the best quality embryo or subsequent chick. So we get a little bit further out, 14 days out, you get out here, over 2 weeks, we're looking at 30 to 40% of those eggs that are fertile are going to die early embryo mortality. So that's why we see it. And I know everybody, and I talk about this all the time, and we all know that and have seen it. I don't know whether we put 2 and 2 together on it as much, but when our fertility starts to drop in our flocks, majority of the time it's due to less frequent mating. And when we have less frequent mating, our fertility is going to drop, and we're also going to see a rise in early embryo mortality. See this a lot in older flocks. A couple reasons why we might have an increase in early dead in older flocks. This is one of them and the primary reason. So we just have to be aware of that. And so does that only affect the chicks? Not necessarily, but kind of going back to this is this chart here. So we use different doses. And if we're using a small dose of sperm, we're getting down to these fewer sperm holes in that site of fertilization at 6, 8, 10 days. That's why that chart I was showing you, that picture I was showing you, it had like 2 little sperm holes, tells us we have infrequent mating. Could still be fertile or not. But when we look at a collective, or we look at an egg can be good fertile or sort of fertile. What's sort of fertile? Well, that particular egg is going to be, an specific egg is going to be fertile, like we see here, till a point in time where it's a binary scale where it's completely switched. Okay, it's not fertile. And so we know when that happens, but we also have this time here that yes, we have fertility, but it's not necessarily the best fertility because we're getting a lot of early embryo mortality. So we look at an individual egg, it's going to hit a point where it's no longer fertile. Collectively, that could go 20 days, 22 days, 24 days. We could still have some fertility in there. But anytime we're getting out that long after a single mating, we're asking for some problems. When we looked at the effect in the hens themselves, and so I talked about that old stale sperm effect, sperm storage in vivo, meaning in that hen, when inseminated hens and collected set eggs from 0 to 5 days after a single mating, and then 6 to 10, 11 to 15 days. Of course, we expect our hatch to drop when we get out, less frequent matings. So even those that are fertile, we get a lot more embryo mortality. Hatch of fertile is down over 10%. That's our embryo mortality issue right there. And then when we also try to grow these out, we see the same scenario growing them out. When we're looking at eggs that the hens after a single mating, we're talking 2 weeks, 11 to 15 days after a single mating. There's still some fertility there, but we raised the broilers, grew them out, and we saw a smaller 0 day or day of hatch body weight. 7-day body weight was less, 14-day body weight was less, and the mortality was significantly more here. Again, the chicks are not as viable as they should be if we have more frequent mating situations. So we just need to kind of remember those things as we go into some other stuff. So what does it mean? It says by ovulation, there's 1 day of development. And so we've got to take care of that embryo. The storage length and temperature that they've been stored at can be have an influence on hatchability. That goes into a whole different talk where we can talk about egg storage conditions and parameters. And those storage lengths, whether they be in the hen or in our egg storage room, will affect 2-week livability and performance of broiler chicks. This chart I showed you was from in vivo storage. We also did the same thing with just storage of the eggs and then pulled them out, and we saw the same scenario. Again, chick is not a chick, and I want to stress this a lot with people is we're often always looking at how do we get the best hatch? How do we get the best hatch? How do we get the best hatch? And I was like, we need to pay attention more to just the best hatch and say, how do we get the best quality in our hatch? Because I can tell you right now how to get a little bit better hatch, just leave them in there longer. But that's not the answer because you're affecting chick quality. We want the best chicks that are there. As long if you leave them in too long, you're going to lose the first of that bell-shaped curve. So we've got to make sure we're really focusing on yes, we need quantity of chicks, but we definitely can't ignore the quality as well. And a number of factors will affect that. So fertility, egg handling, not going to talk about that here, but the egg handling starts at the farm and continues until the eggs are set in the incubator. That's essentially everything that happens from the time that egg is laid. If it's stored at the farm for several days before it goes to the hatchery, That's all that point count in some places, take them directly to the hatchery that same day, but essentially anything that happens before from the farm until the eggs are set in the incubator, we call that egg handling. So that's temperature, humidity, the roughness, if there is any, of which are handled. We see some places with really rough roads coming into facilities, and that is going to have an effect on our embryo livability and our hatchability. So all that is considered, we consider that egg handling. What's happening in the actual development of the embryo? I'm not doing a whole thing on embryo development, just the nature of time. I mean, I've talked about it enough in different places. I believe I've talked about it in previous webinars. If you really want to look, you can go back and look at that. But during that that development process, once the egg is fertilized and we start seeing embryo development, we have a procedure called cell differentiation. Essentially, that's where certain embryonic cells are relegated or assigned to become different parts of the chick. I'm simplifying this very much, but essentially we've got fertilization, we've got an embryo starting to grow. Cells are going to say, you're going to be ectoderm, you're going to be mesoderm, you're going to be endoderm, you're going to be part of the brain, you're going to be part of the heart, you're going to be part of the kidney. And so although that is occurring at this very early stage in the first few days of the cell differentiation, we need to remember that this is a biological function. And in biology, the phrase I always use is you never say never, never say always, because there's variation in biology. There just is. This does not always happen in the exact way it's supposed to. That's where variation comes in. That's where our mutations come in. Some of them, when we're dealing with typical, just like color mutations or different things, if you follow different animal species at all, you know, we as humans take advantage of that. In nature, it normally weeds those out. But this is one of the ways that we see this differentiation. If this is not occurring right, then we see a point in time where we see some embryo loss. So looking at our embryo growth, differentiation is considered up to about 10 days. And really what I was talking about is that the parts that are going to endoderm, mesoderm, whatever, are really occurring in the first few days. But we still have some cells that are being relegated to different areas after that. But then once we get out to 10, 11 days, then we hit that growth phase. And that growth phase is like, okay, all that's done. Now we're just going to grow a bigger chick. So when we're looking at our residue breakout, we need to say, okay, what the differentiation part, is that greatly affected if we have a 2 or 3 day embryo loss? Is that greatly affected by how our machine is actually running? Or is that more an effect of previous conditions? It's usually more of an effect of previous conditions. The growth part, that's the incubators and the environment we're given the eggs. So when we see, and I'll talk about that later, when we see some losses late in production, that's when we really start focusing on the incubator itself and the conditions. So looking at the fertility itself again here, understanding that fertilization process and what occurs before we actually get a fertile egg. So looking at embryo diagnosis, and again, I've talked about this before and in much more detail. As a matter of fact, in 2021, we did 2 separate webinars on 2 different phases of residue breakout and incubation and evaluating chick quality. I encourage you to go back and look at that. I believe it was 2021 we did that. But bottom line is that you really should have a lot of people that have direct knowledge of the breakout results, but also there should be more people than just 1 or 2 that are actually doing this. I mean, I go into hatcheries a lot and I monitor this. Normally I go in and they say, okay, you want to start training us on this? And I'm like, no, not really. But I'm going to sit here and watch you do it for a while and see how you normally do it. And it surprises me sometimes where you have 2 different people at the same place that don't call things the exact same or don't even record them the same, don't understand it. So I think it's best if managers or assistant managers or whatever understand the process and are there periodically to observe the breakouts. How do we record this data? I think this is where we see some things, we definitely see some things get lost. This is the sheet I use. It's not the only sheet we can use. It's the one I happen to use. It works for me. I've spent many, many years in research situations to where it gives me the opportunity to record as much as I want, but I'll go over a little bit more of this a little bit later. But having a good method of record keeping and recording our data. You know, it just doesn't, and I've had this before where people have sent me a basket of chicks, a photo of a basket of chicks, and they say, look at these chicks, how poor the quality is. What happened? To me, I'm like, well, was that basket just a regular basket that came through and there's that many bad ones? Or is that the result of 100,000 chicks hatched today? And that's what you have left. So we need to know, you know, percentages. And then we need to chart this. We need to look at trends and stuff over time. So it's not enough just to have a photo and a visual. We've got to gather data and look at trends, chart it, and you know, as best we can. And we'll talk about a little bit more of that later, but we've got other webinars where we could discuss that as well. When we troubleshoot hatcheries, first thing I always do, how can the problem be identified? Is it a specific flock or flock ages? Specific setters, hatcheries, this is a big one. You know, I mean, we have people that says we're getting poor hatch. They can identify, and an example I use is in one hatchery, and they were having some issues, and they identified it. Well, it always seems to come from this one machine, the same machine all the time. But when they hatch out of multiple machines, it kind of gets mixed up, but they're able to track it back and say, okay, now we know we've got an issue with one of our setters. It was poor ventilation. It was not ventilating properly. It had massive hot and cold spots in one machine, which caused a lot of embryo loss. And once we get that embryo loss, and it's like, oh, it looks like it happened overall. Really, it was just like isolated to one place. So is it specific setters or hatcher? Weather patterns. I mean, heck, it's freezing cold where I'm at right now, and I guarantee you, and I know this because I've seen this, is that people that have breeder farms, if it's, you know, if it gets down to where it's, you know, -10 Celsius or -15 Celsius, we've had that in some of our places, cold, they're not going to be picking up their eggs as often. They're just not. And we will see that 3 or 4 weeks down the road when the eggs finally start trying to hatch, those that they left in the farm longer. And so our handling was off. Seasonal changes, is it routine? Recent changes in management practice or personnel. That's another one we need to track. Does the problem persist? And I've been caught many times not asking this question, and I really wish I should have, where we've been on a remote call with customers and they bring up a situation and they say, hey, we're seeing this. We've seen chicks like, you know, gave an example. And so we talked about it. And I was like, well, how long has this been happening? And they said, well, it really only happened one time about 6 months ago. We've never seen it since. Okay. So we've wasted a lot of time talking about this because, again, biology, we have variations here and there. So does it persist? If not, okay, we need to just keep watching it because we don't know what happened. What is normal? What should be expected? And as people are switching bird combinations in the past and the management they use, we have to be cognizant of that as well. What is normal? And before I get into actual breakout on here, what is normal? This is a biological system. Remember what I said a little while ago? You never say never, you never say always, because it's a biological system. There is going to be issues. Fertility, 100%. We all want 100% fertility. You know what? It is not possible. And I've had in my history, the universities worked with a lot of smaller flocks, so they're not commercial and they're smaller flock, just doing things on a small scale. And I hear them talking, so I get 100% fertility all the time. Well, no, we don't. I mean, you can go all through the animal kingdom, and you know, horses, dogs, whatever, people that are breeding different birds, and they never get 100% fertility. There's always some that just don't work. Hatcher fertile, 100%. That's not possible either, because like I talked about, the developmental periods when we don't get proper cell differentiation, proper growth of that embryo, we don't get that, we're going to get mortality. Hatchability, 100%, it's not possible. Chick quality, 100% perfect, it's not possible. So we need to understand that. So what is normal? Fertility, 1 to 2% infertile, I think, is going to be, you know, that's about the best you can do. Depending on breed, strain, flock, health status, whether you're natural mating, artificial mating, you still should be in that 1 to 2% infertile. That's just a normal, given a big enough sample size. I'm not talking about a small one flat of eggs, but a good sample size. Embryo mortality, 4 to 5% total embryo loss. And this is when our birds are doing the best they can at peak production, where we got our best fertility, hatch, and everything. We're still going to get 4 to 5% total embryo loss. That's split between early dead primarily early dead and late dead. Some mid might be in there. We typically don't see that as much. But I like, so that's one thing in my sheet, I like to track that. What is our total embryo loss? That tells us something too. And that's normal. 4 to 5% total embryo loss is normal. That fits in with our embryo development and the problems we have with that. So again, now we're back to this hatch residue breakout sheet that I shown before. Again, like I've liked this one. I've used it for years and years and years. Many of our customers use it or something very similar, where I can track. You can even track in much more details eggs that are small end up, or cull eggs, or early contamination, cracks, transfer cracks, whatever the case may be, anomalies, exposed brains. I like to have the ability to track that all. It's not necessarily important for every egg to record everything, but what we have to do is we have to focus on what the most important things. The most important parts is infertile, our early dead, our first week mortality, and our last week mortality. Those are the three big things that I follow. And in that last, we got to remember, in that last stage, 15 to 18, 19, 21, we have a transfer process occurring right in the middle there that can also affect mortality here. How do I differentiate? So when I have these little categories here, I've tried to make everything as simple as possible. So what are the key factors I'm looking for? Fertile egg, donut-shaped germinal disc, and then we have other factors we look at: eye pigmentation, egg tooth, and I'll talk about each of those things. Again, fertile versus infertile egg, we've got to be able to determine this. This is a big thing, and I know Dr. Bill will talk about this as well, but we really need to know if we have a fertility problem or not. Because if we, and this is very interesting going into hatcheries, most of the time, if I take a group of eggs and ask people, is this fertile or infertile, and some of these that might be in between here, just the nature of the business we all work in, most people on the breeder side are going to say, yep, that's a fertile egg. The hatchery messed it up. Most people in the hatchery, if it's an in-between, they're going to say, nope, that's infertile. You as a breeder group send us an infertile egg. Okay, we can get into that quabbling if you want. There are some in here that are in the middle. That's why we don't just do our fertility assessments on unincubated eggs. But we all have different areas of the business we work. But knowing the difference between truly difference between an infertile and a fertile egg will tell us. I don't like to go and say we just are having a horrible hatch, and they said, oh, we got all this early dead, mid dead, and so then you go and start looking at it and realize it's really not. It's a fertility issue. Well, that's largely out of our hands. But if you miscall that, then it kind of sends you chasing down the wrong road there, trying to troubleshoot. So I talked about the things I look for. The pigmented eye. We're looking at an embryo here, and this is not a nice little embryo. This is an embryo that died, in that first stages. A pigmented eye is going to occur about day 4 of incubation. We're going to look for that pigmented eye. If it's pigmented, then we're going to put it into the appropriate category. Egg tooth is going to be prominent at about 8 days, and I'll have another little area. I'm going to show this up with the numbers so you can kind of see it a little bit better. But about 8 days of embryo development, we're going to see that egg tooth. Remember what the egg tooth is? It's that little tooth on the end of the beak that they use to hatch, to get through the shell. The little area there allows them to break through the internal membrane into the air cell, and then they peck through the shell itself to get out. But this is prominent at by 8th day. After they hatch, it's gone. It has one function, one purpose to hatch. Once they're done hatching, then we don't need it anymore. They don't need it anymore. Day 15, we start getting prominent down covering. This is probably the one that's a little bit, objective, subjective. You can say, okay, is that prominent? Is that not? But you can kind of tell, and we don't get a lot in that 14 to 15 days. But at 15 days, prominent down covering, yolk withdrawn into the body. Most of that yolk will be withdrawn into the body at about day 19. So when we look at this chart here, these are the things I look for right here. So if we have, fertile or infertile egg, where is that going to go? Okay, it's fertile, it's not. If it's infertile, categorize it there. If it's fertile but died, we'll categorize it right here. What about the day 4 eye pigmentation, prominent? We see that eye on that embryo. We say, okay, it reached day 4. And it's prominent. It means you open that embryo up and you see it and it's like, oh, there's the eye. Okay, it's prominent. Okay, we got to day 4. Is that a big difference in 0 to 3, 4 to 7? Those two? Not as much. I still categorize these all as total early embryo mortality, but it does give us a little bit clue if majority of our dead are early dead are right here, which they normally are. That's primarily an egg handling issue. Okay, when we look at the egg tooth present, right here we see that egg tooth. If we see a embryo, it's dead, we see that egg tooth pops out of us. Oh, there's the egg tooth. It made it to day 8. All right, it's a day 8 to 14. That's a middle dead. Typically don't see a whole lot of those. Shouldn't anyway. Day 15 is when we see the down present. That gets us into that right before transfer. Okay, we've got some chicks here, a lot of down. If we get a lot of mortality here, then we can say, okay, it appears it happened before transfer. Like, okay, so now we can kind of compartmentalize, okay, where do we need to look before transfer issue? And then if we start seeing embryo mortality where the yolk sac is mostly withdrawn into the body, like we see here, that's a 19 to 21 day. Again, that's after transfer. There's a clue right there. You know, if we get everything else is good and we're seeing a lot of late deads, there's a number of things we can look at. We'll talk at a few, but one of them is like, well, we pretty certain it made it through the rest of the process fine. Could have been a transfer issue or a total incubation thing. So that's the categories. This is why I like this chart, because it's like, this is what we look for. I know years ago when I started doing this, it was more like, oh, is that a day 16 or is that a day 6? Well, I'm not so concerned with that. I want them in the right categories here. So I looked at different things that were happening in that bird that we can truly categorize what's going on here. What's the other important thing? And I have actually seen this issue more than I would like to have, because we're just trying to assess what's happening in our hatchery. So it's very important to have good quality data and accurate data. So when I look at this as like one test, this is a test that we've done in places, and sometimes it kind of gives them their answer. If everything should equal, every egg should be accounted for. If we had 168 eggs in a tray, 153 chicks hatched, 5 infertile, 3 0 to 3 days, 1 4 to 7, 1 mid, 1 15 to 18, 1 14 to 21, that all equals 168. There should be no eggs, something lost. So the reason why I bring this up is one, every egg has to be accounted for. And what I've seen is sometimes people will put, yeah, it was a dead pip, and they record it here, but not here. So now you had 168 eggs, and you only have 167 even accounted for, because this dead pip here was never recorded as a late dead. Well, guess what? It's a late dead. It died. If the chick does not come out as a full healthy chick, you can place it. It needs to be recorded up here somewhere. It could be a cull chick. If you have a cull chick, that would add into accounting for every egg that was set. And I've talked with people before, and they've said, well, I think our system, if we count it as a live pip or a dead pip, it automatically moves it up there. Sometimes it does, sometimes it doesn't. They don't know. But that really can skew your data. We dealt with a customer for a while that had 0.75% to 1% lost. I mean, they couldn't figure out, why is it we set these eggs and we always have this loss? And they were doing some of this. They were counting eggs. Oh, we had an exposed brain. That's why it died, but it still died. So every egg needs to be accounted for. Again, making sure we have accurate data, because we don't want to start making decisions on data that is not accurate, because that will lead us in a wrong direction if we're trying to troubleshoot. Looking at our mortality patterns, first 7 days, about 2 to 2.5% mortality is normal. Again, like I said, the cell differentiation is a big factor right there. Most of the time, that's why we have these early mortality losses. that can be exacerbated by poor egg handling, age flocks, possibly incubator problems. I haven't seen that as much, but those first 3 days particularly are typically pre-incubator. And that, so that's what we're looking at. But about 2 to 2.5% is what we consider normal there. The mid mortality, about 0.5% or less, should be less actually. That is normally considered a breeder nutrition problem. And in all honesty, in my years, 25, 30 some odd years, seems like a long time, but doing this, I have rarely seen consistent mortality problems in a hatchery that are consistently mid over and over and over. Being that if your nutrition is off enough in your hens, you might have a one spot where one house got broiler feed or something, and boom, man, it really messed everything up. But if the birds have a diet formulated for them, or they're fed a diet that is not suitable for them, eventually they'll quit laying eggs, because they lay eggs to make sure all the eggs have the right environment and right contents to grow a good chick. If they can't do that, they're going to quit laying eggs. So I don't see that too much. And if you see a lot of it, sometimes I go back. One of the things I've seen a lot where is they have high mid, and I go back there and see, well, some of those were actually day 6 or 7 embryo. They're actually more early than mid, because we really shouldn't see a lot of mids. Late, about 2 to 2.5% in that last week as well. This is where we start getting some incubation problems: temperature, humidity, turning, pull time, flocks, egg orientation, things like that. So when we look at our mortality patterns, this peak we see here at 2 to 3 days is normally due to cell differentiation. That is very similar to if we look in other mammalian species, including humans. When we have incidences of miscarriage in humans, many times that is due to problems with cell differentiation, and the embryo or the young fetus is not growing properly. So it's like, okay, stop. Mother Nature has a way to say, nope, this is not good, let's stop. And that can be aborted. In the chicken, it can't be. So it just sits in the egg and we see it later. But this is something that happens throughout the animal kingdom, is we have this filtering and sorting out of improper growth and development, and it has a way of just sorting it out itself. So 18 to 19, a very important thing happens here to our embryos. So they're growing along, they're cruising. Hopefully we've already transferred them, but it's right about that time, depending on when your transfer time is. But the chick does something very important. It switches to pulmonary respiration, meaning up until that point, all of its respiration was through its circulatory system. And at that point, around 18, 19 days, pushing more to 19, that chick pips into the air cell. The leg tooth air breaks the membrane, pips into the air cell, and then they have that first breath of air. And then now they are getting all the respiration oxygen exchanged through their lungs, through their pulmonary respiratory system. If that doesn't occur properly, we get death. And again, that's another biological thing, is that we're going to get some death. Things just don't go exactly right. Now, is there things in our incubators that can make this worse? Of course. We can, if we've got high temperature, low temperatures, no moisture, a lot of different things like that, we can change some parameters that makes it more difficult. But that is a normal thing that happens as well. And then our last point here is hatch problems. Just hatch problems. Okay, they tried to hatch and they couldn't. Why? Well, maybe the egg was upside down. Maybe the shell quality wasn't the best. Maybe it lost too much moisture because of that shell quality. A number of things could happen there, but the problems are just hatching. They just couldn't get out. We are going to have some of that. So this is the second peak that we have. First peak, second peak of what's normal. And again, like I said, this is generally 2 to 3%, or 2 to 2.5%. 2 to 2.5% here is what we consider normal. When we're evaluating this whole group of eggs, when trying to troubleshoot, I think it's important to know the elevations in what we expect here from that 2 to 2.5%. We can generally look mostly at egg handling. I don't think we need to go and start looking at, is our machines have a uniform temperature in there or not? Honestly, and I've done this before, and I've tried to manipulate this. I've tried to set eggs at high temperature, low temperature. different things like that. And we can increase mortality, like I suspect, but typically it's not till 7, 8, 9, even 10 days. If they're too cool, it might be 10 days before they actually die, but they're going to grow. Hot summer days here in Arkansas where I live, I can take some eggs and leave on my car seat out there for a couple of days, and we're going to get growth. Doesn't have to be perfect temperature, but we'll get some growth. We won't necessarily get a chick, but we'll start getting some growth. So we look at egg handling more than our incubators if we see elevations in mortality in that first week. That middle week, that's where we start looking at nutrition problems. And again, don't go run out and start blaming your company nutritionist. Oh, you're formulating a horrible feed, because if you're getting good egg production, good livability of the hens, the diet's probably good. Long term. Now, we've seen where the wrong feed's been delivered to houses, and that's catastrophic. But long term, if somebody's getting good egg production, good livability of their hens, good shell quality, we shouldn't be seeing elevations in embryo mortality in that mid due to nutrition. And then that last week is where we really start focusing on incubation problems. So when we're doing our residue breakout and saying we're just getting a lot of late deads for one reason, and I'll talk about a few of them, but not very many. That's when we start looking at the parameters in our incubators. So again, the chart, one reason I like to use this chart, very broken down. Anything in this category would likely be egg handling issue, nutrition issue, or incubation issue. So the chart actually allows us to categorize the critical events that are occurring in the development of the birds, and then we can proceed from there as far as troubleshooting. I'm not going to focus on the early stuff. I think Dr. Beal is going to do a little bit of that next month. So I kind of took that portion out of this. If you want a little bit more detail on this, again, go back to a webinar from 2021. I did this. I was quite a bit more detail on it. Presented this at some other places as well. Day 19. We get embryo loss at day 19, signifying it made it to that point. Yolk sacs mostly withdrawn, amniotic fluid's gone, embryo's taking up most of the space. What if we get a lot of losses here? Be rough transfer cracks, delays, if we're not having a smooth transfer pattern. Again, transfer here. Temperature, humidity, ventilation, all these things, upside down eggs, all these things can affect that if we start seeing a lot of 19-day development. That's right after transfer. At 20 days, again, yolk sacs completely withdrawn. They probably at that point have already pipped, breathing into the air cell. Again, if pulmonary respiration was not initiated at that time, we're going to see some loss, and that could be in that 20-day range where we start seeing that. And we could also see some poor shell quality, hatcher temperature, ventilation, time of transfer that would signify or contribute to losses at 20 days. So a couple of these here, I'm going to have you think a little bit. I like to do this more in a classroom and one-on-one situation. If any of you ever heard me give a presentation somewhere, if the numbers are small enough, let's make this a discussion. Because I spent many years as a professor, I like to ask people questions and make them answer or get their answers. So I want you to just think about it. So if you have a lot of dead pips, live or dead, they're dead in the shell, full-term embryo. What could be low humidity or temperature for long periods? Hatchery humidity low, high temperatures during hatching, poor ventilation, prolonged egg storage. So think about it. So if we have a lot of pipped live, we have an excess number, way too many normal looking chicks. They're pipped, they're live, they're just not hatched. We can immediately look back and say, okay, temperature is what gives us our rate at which our embryos grow. We have lower temperatures that we got just too slow growth. So when it's time to pull those eggs, all the chicks, we had some that just never quite made it to that period of time. We pulled them before that point. That's where we start saying, I can say, hey, I can just don't pull them for 8 hours. You'll get a lot better hatch, but the first ones that hatch are going to be poorer quality. So we can't really do that. We need to go and address what's happening in our machines. So we think about it. Pip, dead or alive, what's going on? They're not pipped at all. Dead in the shell, full term, large yolk sac. Again, same type of thing. Setter temperatures could be low. It's just slow. They're just not growing and developing. Never even got to that point. Ventilation, moisture loss. I think we see that sometimes. If they're just wet, they didn't lose the moisture. They try and pip into the air cell, and the air cell's not there, developed as it should be. So now we got too much moisture. They may not be able to switch to pulmonary respiration. That would be an internal pip, but not an external pip, or they may not even get to that point. Egg storage can affect this. Chilled eggs that transfer, that slowed them down. Again, we're slowing them down here. Partially pipped. This again, that's the same thing, is that when we see this, they pipped into the air cell. The CO2, they actually, when they pip, their first breath is a high partial pressure of CO2 in that air cell that encourages them to get out faster. But if we don't have the proper ventilation in our hatchers, and we start getting these pips and they're starting to breathe the air that's they're exposed to, either in that air cell or in the incubator itself, it could choke out. It's basically suffocate because they don't have enough oxygen, so where ventilation's not correct. So this would be, they're starting to pip, they're just stopped, but the embryo's alive or dead, fully developed. What if as a collective group, they're just hatching too early? A lot of noisy chicks, they're thin, dry around the legs. We start looking at setter temperature too high, breed differences, or small eggs. And that is that's something that we could see. That's the science of incubation, or the art of incubation, where we know what we're supposed to do, but we evaluate. We are consistently getting chicks just hatching earlier than we want them to. Well, we can do changes to the profile. We can look at our machines and make sure they're uniform within those machines to make sure everything is growing uniformly and correctly and on time. So if all of them are hatching too early, but they're uniform and they look good and everything like that, typically it's a fix we can do with looking at our profile, temperature profiles. What if they're all late? They're all green, swollen abdomen, unhealed navels, wet chicks? We'll start looking at our moisture loss, our setter temperature, humidity, excessively large eggs, old breeders, stored too long is going to delay the required incubation period. We see a lot of this, green chicks. The art of incubation, we can say, okay, now we know, let's make some adjustments so we can kind of bring these back into line so they're no longer hatching late, they're hatching about when they should. Slow hatch or a large hatch window means you've got a lot of early ones and a lot of late ones. You could have a mixture of eggs, some stored excessively long, some not stored long at all. Old and young breeders could be mixed. Mix of large and small eggs from these breeders, proper handling. Hot and cold spots in our setters or hatchers. If we have a large hatch window, and I think that's one thing I'll really, really emphasize with our Jamesway products, our Jamesway single state products, is we create a very uniform hatching window because we have a very uniform environment in our setter cabinets that will allow the birds to all develop as they should, as long as we're not doing some of this, mixing some long stored, short stored eggs, old eggs, young eggs. We can kind of look at that. Sometimes we have to, but we can address that if we get a large hatch window, a slow hatch. We can look at profiles and machine function. So then just having an action plan of what we're going to do with it. Making sure we have accurate breakouts, make sure we have a standard summary, what's normal, taking that data, analyzing it, and then having some sort of an action plan of correction. I have been, and I've used this example for years. Many years ago, I went to a hatchery and asked if they were doing breakout, and we do it every day. We were told to do it every day. I go, so what do you do with the data? And they go, it's recorded on the sheets they gave us, and it's over there in a file cabinet. They never used it to do anything. They were just told to gather it. So have an action plan. Use the information as a management tool. Gather it, sort it, compile it, put it in graph form, however easier for you to look at it. And then making sure the hatchery manager and supervisor are intimately involved with that so they can know what's going on and things aren't just happening. They kind of know what's going on. So this kind of a quick fly through of a residue breakout and kind of ignoring the first parts of it, I guess. But anyway, if you have any questions, I guess that's okay. How do I stop?
Thank you very much.
How do I stop sharing?
No, we're still. Do you hear me?
Yeah, I can hear you. I'm trying to figure out how to stop sharing it now.
Oh, okay. Because we're seeing your presentation still. So thank you very much.
Oh, there you go. There. Okay. There we
Okay,
go.
there we have you. Thank you very much for your presentation. As always, it is very informative, and we get like an upgrade of how should we analyze all this information generated by the hatcheries and all the residues.
And there's a lot. I mean, I just can't emphasize enough how important it is to use the data we have. And the data is what's happening in the hatchery. And I've said for a number of years, I was like, you can look at a good batch of chicks and say, oh man, these are great chicks. Look how good they are. Like, but what does your mortality look like? They tell you a story too. It's not just the best quality chicks I've seen. It's like, oh, we got a bunch of ones that died, and yeah, these are the condition situations. They will tell you a lot. So anyway, enough of me blabbering.
Yeah, and we have like one question from a customer, and that's something that you touched in your presentation, and it is very important, and is what is normal and abnormal, and how should you compare the data? So should it be like compared with breeder genetic line, or should with a standard line? So how should you compare the data?
Yes. kind of both. And that's why I talked about in there as having a baseline. What is normal? And not only what is normal from the primary breeder, they might tell you, says, this is what we normally see. What is normal for you in your operation? And I think we need to know that. But, sometimes we have people, they don't do certain measurements in that hatchery until all of a sudden there's a problem, and then they start doing them and they're not really sure what it normally was. So they don't know if something changed because they didn't know. So both, the answer was yes, both. Primary breeder recommendations, your history. A lot of big companies will have flock history. Last week, this flock did this. This was our breakout. This is our fertility. This is our livability. And the week before, and the week before, and the week before, and they develop a standard of what they expect themselves. And it may be different than what a primary breeder tells them, because they can do better, or maybe not. But you need to know what's normal. So incorporating all that.
And maybe you have already answered this, but let me ask you this: is there any standard for GPs or GGPs? Or should they compare it with their historical, and how should they analyze those data?
They're definitely different. They're definitely different. And having worked with primary breeders for a number of years, we try and give, so if we give a profile for a particular genetic strain, that's what we have found that works with our customer base to give us the best results. Well, if you go to that genetic company and say, well, what are you getting on your GGPs? Well, they don't compare. It's different genetics. It's not the same that you're selling to the customers. Oftentimes they'll tell me that, oh yeah, this particular line with ours, we have to incubate these a little bit different than that line. But once they get down to the parent stock that most of y'all are doing that are growing birds, they're more uniform. They've all got the same genetics, but up at the top tiers, you've got GGPs, male from this line, female from that line. So it is different. And typically, from what I understand, is the hatch is typically not as good, partly because sometimes they got to store eggs for over 3 weeks to have enough to meet a need. So of course, we're going to have something different. So the genetic companies will give you good advice on what they see, but I wouldn't necessarily say, no, what do you do with your pedigree birds or GGP birds? Because they don't necessarily correlate with each other exactly.
And from this, the same topic, we have a question. Do you have like a standard related with malpositions and abnormalities, like deformities?
Yeah, we do. I mean, typical. I think most people kind of say, oh, we need to stay under 0.5%, depending on the age of the flock and everything. There are some little bit differences, but I think most people don't get too worked up about it as long as we stay under 0.5% overall long term of each of these abnormalities. But again, if you get a lot of malpositions, you could have a lot more malpositions. That could be because you got upside down eggs or some other issues in there. But typically, we're never going to see where we have none of those, no malpositions, no exposed brains, none of that. You're going to always have, it's biology. You're going to have some. So we want to keep that under that 0.5% and plug along there. Don't get too worked up over that. Unless one of those areas really flies up. All of a sudden you got 3% exposed brains, and okay, so now that's an issue. But a few of them. I was in a hatchery one time, and they brought me a flat of eggs, and they said, "This is what we're seeing," and all the eggs in there were all malformation. You know, they had multiple heads or something like that. And it happens like, man, that's a lot. And so I said, well, where did this come from? And they said, that was from today, like the whole day. And I go, okay, well, that's not an issue anymore. You hatched a lot of eggs today, you know, but if it's like, yeah, we're getting this every single time we pull a flock, then that's an issue. So
Yeah, and actually that is a good direction to go through, because we have people asking what should be the size of the sample? Where should they sample the eggs? Where should they take out the eggs from the machine, from the special flock? How many trays should they sample? What will be like a good representative sample?
I'll just say most people will use a 3 basket sample. Some will use 4. But everyone says, what's best? And well, let's look at statistics. I go, 2 baskets is better than 1, and 4 is better than 2, and 25 is better than the 4. But that's not realistic. So most of them settle in that 3 to 4. The other thing is I think there's 2 reasons why sometimes we like to say 3 baskets, but not just 3. We want top, middle, bottom. Because with this chart, what I've done with this chart is I've gone in hatcheries before, and we collected a lot of data as in a multi-stage hatchery. And we had all this, so okay, let's look at all the 8 flocks we broke out today, and look at all of them in the machines, and all those that were in the top position, in the middle position, and the bottom position of their machines, regardless of flock, and seen like 8 or 9% difference between late mortality on the bottom all the time, regardless of flock, as opposed to the top. Okay, so normally we do top, middle, bottom to get a uniform sample to measure that flock. If we track that on those that were top, middle, or bottom, we might say we are consistently seeing a problem on those that were on the bottom, or those at the middle of the rack. And so we can use those 3 or 4 or whatever we want to use in position wise and see, is this a consistent problem position wise in our machine? So 2 reasons why we want to do multiple samplings.
And how often should people do an embryo diagnosis or a residue breakout?
That's a personal thing, and a lot of it is determined by economics. Unfortunately, you know, there are some areas where labor's cheaper, and it's like, hey, we can do every flock every week. Great. A lot of places like, no, it's not worth it. So what they'll do then is they'll put the flocks like on a routine. It's like, okay, as long as they're hatching good and everything's going good, they might do every flock every 2 to 3 weeks. They might do a residue breakout of the flock. Sometimes 4 weeks I've seen, even in peak production, because they're going well. And then as they go on, you start seeing problems. You might start doing them certain flocks every week or every other week. Might start doing them more because they become an issue. It's purely what works for your system economically and how much time and labor. It's definitely worthwhile to do all the time. Some will do more of it just to get a baseline and only chase those that have a problem. Some will say, we're going to do more flocks more frequently. And that's fine if you can do that. It's all good data.
Right. And there are a lot of questions about contamination, like general contamination.
And I'm like you and I talked about before, some of those types of questions we can respond to you directly, because a lot of times there's other questions that we want to ask before we can try and answer that.
Yeah, I mean, there is a lot of concern about contamination, and I think that's a global concern on the hatchery world. It is a lot of worries about it.
We've had last year, we had a hatchery biosecurity discussion on that, because that is a contamination issue, is whether it's coming in from the eggs or it's in the hatchery or whatever. But yeah, that's always an issue. But we're happy to entertain those. Probably better to do it one-on-one in an email or something, so we can get more data and information.
There is another question. Do you find, or have you seen in your experience, any difference between a residue breakout from a single stage compared to a multi-stage?
Yes. And I've had a chart with that, and it's not in this thing where we've talked about that over time. Typically, what we see, I mean, and I'm not just saying this because like, oh, single stage is the best. I mean, I came from the academic side, and when single stage first started being promoted out there, I go, man, if it's done right, this is it. This is the way to go because we have more control over the whole cabinet. So in that sense, yes, if we look at it multi-stage, what do we expect? Our multi-stage work pretty well, but we typically have a little bit bigger hatch window because we don't have as uniform of an environment in the hatch cabinet or the setter cabinet because we're having eggs that are 2 days of embryo growth and 12 days and 17 days before transfer. We have them at all different stages in there. So we don't have as much control. So we typically will see a little bit more mortality. The single stage, the reason why that's so good is we can look at that and say, hey, we're getting a lot of later mortality. Okay, so we can change the whole profile in that machine for the next time, and we can address that and correct those and bring those back into. So typically we do see one better quality check from our single stage. Two, we typically are going to see less residue embryo mortality as it relates to the incubator, not necessarily the egg handling, but yes, we'll typically see better results from our single stage because we just have better control of the environment, bottom line.
Great.
And we have better control of the environment and the egg pack. I'll put it that way too. Like we all the eggs going in machines should be very similar in a single stage, and we know what's going in there. Multi-stage is like, you got all different ages, and then you try and work through that machine to get uniform. So.
And here, like a question about breeders, and it is, which is the best time or how long should the sperm be inside the hen? I mean, how often should the turkey mate the hen in order to have a good number on fertility?
Typically, what I understand, whether it's under artificial insemination or whatever, and some of the work I've done is that if you get out beyond once a week of fertilization, whether it's natural or artificial, you're going to start seeing some issues. Turkeys can store sperm up 4 weeks or longer. Some of the data is ridiculous. It says 40 days they can store. They can actually get fertile eggs still. They're not as quality. And chickens, what I've seen, and particularly broilers, is 20 to 21 days, about as long as you'll see. That I always look at it and says we need to do that, once a week. If you're doing artificial insemination, I would say I would never go more than once a week. During peak in production, when fertility is the best, possibly you could probably go out to maybe 10 days between, but I wouldn't recommend it. I'd say if you're going to do artificial insemination, you need to do it at least weekly. And then the broiler, that's what we encourage, trying to encourage movement and motion through that housing. So even in their natural mating, we get frequent mating. The thing that we remember with our breeders when we're doing natural mating is if you have a set number of males and a set number of females, if you add more females to that, the males typically don't mate all those birds less frequently. They typically mate the ones they like the most, and they'll just ignore some of the extras. Okay. I mean, if one rooster's got to breed 15 hens, he's probably not going to breed 15 hens. He's going to breed his favorite 7 or 8. And that's just the nature of it. And so we can't really say with that. We've got to make sure that we have enough in there because they are selective in who they breed and stuff. And that's a whole other ball of wax that I've done some research on watching, is what makes a rooster know a hen needs to be mated or not. And there's a lot of theories and thoughts on it. I have a bunch of them, but I'm going to skip them right now.
Yeah, actually, we know you like a lot of birds and wild birds and seeing, like watching wild birds. So it happens the same, right? According to colors, they'll try to choose the most beautiful male.
And it's a little bit different in our broilers because they're all white. They'll look mostly the same. But I will tell you, and I have hundreds and hundreds of hours of video recordings in breeder houses of watching mating activity. And it still boggles my mind why a rooster can be cruising along and all of a sudden he stops, he looks up and he takes off and runs past 15 females to get to one that was just standing there minding its own business and mates her. What did she do? did she give us it? We don't know, but there's some cues they're getting that they know who to mate and who to not. And I don't know. And we have some ideas and some other research has been done, but you know, with our, especially with our broilers, they all look the same. They don't have extra colorful feather colors for courtship behavior. They don't have it. So
Well,
we've
who
changed the
knows?
dynamics of them.
Maybe they see their legs, if their legs are beautiful or not, and who knows, their
Yeah,
eyes.
they bat their eyes or something, right, or something.
Yeah. Um, we get another question here. And it is at pool day, if we have like green chicks, is it okay to turn them back to the hatcher or maybe increase a little bit the temperature? Do you think increasing temperature will hurt some other chicks when we do that, those kind of management?
It can sometimes attempt. Typically what we see, and we've been in hatcheries and they start pulling them and they start looking at these are just not ready. The typical and the best response, I think, is just put them back in. Just put them back in and give them more time. Just give them more time. Now, if you're really crunched with time, it's like we can't wait 6 more hours. All our people are here or anything. I mean, you could do a little bit of temperature increase. Got to make sure your ventilation's working good because the fear is always there of getting them too hot, getting them stressed, heat stress. So I don't like to do that. Typically, it's just more time because we've found, you know, our profiles we have in our setters or our hatchers are set, and we found those to be successful. So if they're not, we typically don't want to start messing with the profile as much as we say, okay, we need to give more time. Where you go from that is the next time you set eggs, you just start setting a few hours earlier to compensate that for the next time, rather than start changing the profile to speed them up.
Right, and that happens frequently. I mean, sometimes could be calibration or, you know, like some other issues. So,
Right.
so it's good to like keep an eye on that machine if that happens. Do you see poor performance on farm on the chicks that hatch earlier compared to the rest?
Earlier, as far as like a collective group, it's a shorter incubation period. Is that what you're talking about?
I think the one that hatch like earlier in the hatch window, they refer to that one.
Oh, okay. If you've got a really big hatch window, of course. So you know, our what Mother Nature says, hens clutch her brood is going to hatch within 24 hours. That's what we consider normal. If you start getting to this 35-hour, even 40-hour, we've heard from the first chicks hatch to the last, those first chicks are going to be dehydrated or headed that way of dehydration because they've just been there too long. So you're going to have poor performance. You're going to have more mortality with them. They're going to have a harder time getting started because typically it's like, we know what happens if you got, everyone likes chick weight. Well, if they're dehydrated, they're not going to have the weight and they're going to start off a little bit behind. So yeah, that's why hatch window is so important, is that we want to reduce the extremes from the beginning to the end. You know, even if you say, I'm going to pull these early because I'm worried about the front end, you know, the first hatching ones. Well, if you pull them early, then the last ones are green. They might have open navels, so they're not going to perform as well either. So, you know, going back to old school, small flock people, even to today's age in smaller countries where they have a lot more labor and equipment's different, I've seen them pull part of a hatch and then put the other half back in the same hatcher and pull them like 12 hours later because they got poor uniformity. They know it, but they also know that if they don't do that, they're going to have very poor performance on the farm because you've got some dehydration issues. So they actually pull them twice. Do not recommend that, but in a situation, I've seen that happen, and that's they've just made the best out of the situation. So,
well, we will just answer a couple one because we had a lot of questions there. There is a lot of participation from our audience, and we will get directly in touch with them. And email them their answers because they are like really curious about a lot of topics. So maybe we will go directly to them. Thank you, everybody. Thank you, Dr. Bramwell, for sharing your knowledge and all your experience with us. It is very, very, very important, and it's good to have your opinion and all your information.
I always enjoy being on this side as much as being on your side, but next time I'll be on your side of the screen. So.
Okay, you do great both sides.
Yeah. Mix it up a little bit. It's fun.
Yeah, just a little bit more dynamic, like mixing everything, leaning one
Yeah.
side over the other one. Um,
Yep.
and hey, everybody, just remember we will have another webinar on February 14th. And for the ones who were like, we were a little bit struggling with translation, we did record the Spanish translation. We did have translations just a little bit late, but we did have Spanish translation for the Spanish speakers. So I think we will send that recording as well.
And if you have questions about any of this, I mean, webinars at jamesway.com, you can email that if you didn't get my email in there. Mine's keith.bramwell@jamesway, but you can always contact us there with anything, any needs or whatever. I mean, we're happy to always go out and work with you, whether it's on site on your facility or doing remote contact and trainings, which we're doing quite a bit of as well. So yeah, just let us know what we can do to help.
Great. So thank you so much. And everybody had a good afternoon, evening, whatever your time zone is.
Whatever it is.
Bye.
All right. Thank you. Bye.
Thank you. Bye.